Without further ado, now that we got the recording started, I'm going to go ahead and introduce our speaker for today. So we have Dr. Kathryn McDougal, is a clinical assistant professor in the Department of Biological Sciences at Towson University in Towson, Maryland. Our focus is to provide undergraduate students with the opportunity to participate in creative and supportive authentic research experiences within their undergraduate courses. She was an undergraduate researcher herself at Alma College in Michigan, earned her bachelor's degree in biochemistry, then went on to graduate from Johns Hopkins University of Medicine with a doctorate in human genetics and molecular biology, and did a postdoctoral fellowship at Johns Hopkins Bloomberg School of Public Health in the Department of Epidemiology. She also plays an active role in supporting student success by coordinating academic advising in her department and working to support first-year students and new academic advisors in her college. So today we have Dr. Kathryn McDougal and also her colleague, Akeem Lawrence. So without further ado, I'll pass it over to you.
Great, thank you. I'll go ahead and share my screen. Awesome, okay. So hello everyone. Today I'm going to talk to you about my experience working with students in the Researcher Workbench, share some of my tips and challenges, and also highlight some of the user support resources that you might find useful. Okay, so I work specifically with undergrads, and I use the Researcher Workbench in a course environment, but whatever student population you work with, there's going to be some additional challenges. For example, students are busy. So for me, I work with the students in a 4-month semester, but these students also have other courses they're taking. They're involved in clubs and activities, and also they're involved in college life. I also find that the students are sometimes intimidated to tackle the Researcher Workbench. Some of them have no previous experience in research at all. Some of them only have wet bench lab experience and they haven't done any kind of computer project. And so they might not know much coding, so they have limited skills and they can feel a little lost starting out and just kind of uncomfortable in the research space. So that's something you have to consider. There are also challenges for the mentor. So you yourself have to learn the workbench so you can teach it to the students. And also getting students access to the workbench, there can be some challenges in that. I recommend developing content to teach the skills the students need to use in the workbench before throwing them in. So things like experimental design, which helps make picking cohorts and concept sets a little bit easier. And also brushing up on some coding skills, either R or Python, whatever you plan on using. You need the time to prepare for all of that. Plus, for me, I also teach other courses at the same time, and you need support for that. And so for me, it's departmental and college support. It might be different for you, but this support can be monetary, it can be the gift of time, or just, the positive reinforcement and support of the people you're working with. So I do a semester-long undergraduate course with students, and that course is in a CURE format. If you are unfamiliar with CUREs, they are course-based undergraduate research experiences, and students perform research in a classroom environment. But what makes this different from the traditional cookbook lab is that the students design the projects themselves. I have the students work in pairs or in a set of 3 if I have odd numbers, and the students design and execute the project as a group. The research is novel and relevant, and I make sure there's iteration. And so if your code doesn't work, you have to keep working on it till it works. But even if it does work and you have this beautiful graph, it might not be the right kind of graph for your data. So you have to look at it and think about it. And you might want to change some things and run it again. Or maybe, you're using age and it's a quantitative variable and you want to look at age groups. There's that idea of iteration. And we have time to do that in this kind of course. We don't have to, finish this lesson and hop onto the next thing that's in the lab notebook in this kind of course. If you're interested in learning more about CUREs, I was strongly influenced by the Auchincloss paper, which I've cited down here below. And I kind of went through what their components of a cure and made sure they matched up with my course. My course specifically is called Genetic Analysis in Medicine. I've offered it 3 times, and I'm going to be offering it again in the fall. And so at Towson University, we have a lovely new science building with this big white lobby with this really huge screen. And for some of these specialty courses like this one, we use the screen to advertise our courses. And so you can imagine this big black slide in this big white room on a huge screen. It really pops. And there's a couple reasons I advertise this course this way. You'll notice there's a really big computer on it. And the last line of the course description is that the course includes R programming, data cleaning, and statistical analysis. I still have students that come into class on the first day shocked that it's not a wet bench lab course. So I try to do this so the students feel like they know what they're getting into when they take my course. I also make sure I have the little All of Us banner on there. So if they're really interested, they can Google that and find out more about the All of Us research program. So my target audience for my class are students interested in bioinformatics, genetics, medical research, or even clinical medicine. Usually they are juniors and seniors, and it's just the way our majors are structured, that's the time they get to take that course. I have had a sophomore before who's done well. The prereqs are a genetics course, which for us is an upper level genetics, and a college statistics course. I meet with the students twice weekly in 3-hour blocks. And the students use their own computers to perform the research. That's worked really well. If a student doesn't have a computer, I do have department computers available, but I luckily haven't had to use them. I utilize undergraduate learning assistants. If you're unfamiliar with them, they're basically undergrad TAs. So we do not have a PhD program in biology at Towson University. We do have a master's program, but those students have other responsibilities. So it's really common in our department to utilize undergraduate students in teaching assistant type roles. I chose to work with All of Us because of their commitment to diversity. We have a really diverse student population at Towson, and I knew that this would resonate well with our students. And I really wanted to work with someone that was excited to work with undergraduates. And so All of Us's commitment to non-traditional researchers was really attractive to me. The $300 credit didn't hurt. That means no lab fees for students, no kind of figuring out what departmental pot the money was going to come from to make this class work. And so that was really positive as well. Now, you'll notice my title of this slide is A Grand Scary Experiment. That's because the first time I taught this course was in spring of 21, which was during the pandemic. And so my planning time for this course was during 2020. So as I'm sure all of you know, there was a lot going on in 2020. I have 2 elementary school students who were doing online elementary school in 2020. And I was also redesigning in-person wet labs to be online courses. So when January came around and I was about ready to start the spring semester to teach this course, I was not as prepared as I had wanted to be. And I shared this story because, I sat down with myself and I said, okay, so this is a new resource for everyone, right? Not just for you and your students. This is, I think it was less than a year old by then, the Researcher Workbench. And the team can correct me if I'm wrong. So it was new for everyone. And we are going to learn with everyone else. This is authentic research. This is a real representation of what research is for students. And I share that to kind of express that it's okay if you don't know how everything's going to go. This isn't that kind of course. And these aren't those kinds of projects. So I taught this course in 3 versions. And these versions are different in the types of data they utilize and even the types of tiers that they are. And so I want to share these 3 different versions because depending on what your interests are in working with students, one might work better for you than the other. So spring 21, genomic data was not yet available. So genetic analysis in medicine became data analysis in medicine. And we were completely online due to the COVID pandemic. And I will tell you that worked beautifully. Everything with the All of Us Researcher Workbench is online. We utilized, the big Zoom room for when we had big conversations, the breakout rooms when the students were working together. I would absolutely teach this course again online. So the Workbench works great in that kind of format. I had 2 undergraduate learning assistants, one in biology and one in computer science. The biology one helped with experimental design, and the students have to write research proposals as part of their midterm. And so she helped with that. Computer science, I had Hakeem Lawrence, and I've been lucky enough to keep him as a ULA through all 3 iterations of this course. The students choose what they want to look at. I tell them if it's in the All of Us database, great, you can use it. It's completely up to you. And you have to sell your design to me in your research proposal. And this group chose from the EHR data, the electronic health record data, survey data, physical measurements, and wearable data. We did learn this semester that the EHR data is a little more complicated and the students need a little more time to work with it. And the projects were really inexpensive, $20 or less per project. And honestly, thinking back, $20 might be a high estimate. So I just want to touch on this slide briefly because I'm so interested in what the students come up with for their projects, because it really is driven by them, and it really it helps me recognize what they're thinking and what's important to them. This semester, COVID was super important, obviously, because we were in the middle of the pandemic. I highlighted the COVID projects in purple. The COPE data had just come out, the COPE surveys, I think in December of 20, certainly in the fall of 2020, they came out. And so half the groups did a project with that data. I also had a group that really got into the survey data and did an HIV and access to healthcare project, and 2 groups that braved the electronic health record data. And they didn't get as far along as in their project as they wanted, but that was okay because we actually learned a lot about the electronic health record data. All of these groups made it to a poster, which is part of their final project. Because of COVID, they weren't able to share that poster with the campus community, but they all were really proud of their work and they felt it was a really fulfilling experience. And so this is an example of a register tier kind of project that doesn't use any of that genomic data. In spring 2022, I taught this course again, and I heard genomic data was coming. I didn't know if it was going to come the first week of classes or if it was going to come the week after the end of the semester. I had no idea. So we kind of started out with version 1, and if the genomic data came out, then we would use it if we could. I believe it became available at the end of March. It took us about 2 weeks to figure out how to access it and actually use it. So we did end up using some of the genomic data, really only a handful of SNPs because that's all we had time for. This semester, we were completely in person, and that worked great as well. I had 1 teaching assistant, Hakeem. And again, the students picked from a variety of data types, and the genomic data we used was the genotype array data. Now, to access the genomic data, we had to leave the world of R for a little bit and figure out how to get the data out based on what we wanted to do, and looking at the sample code available in the user support section. We thought using Hail would work best for us to pull out what we needed, save it in our workbench, in our bucket, and then move it into R. And there are ways to save the files and convert the files so you can use it in R. And to do that, we had to use Python. I don't know Python at all, but Hakeem Lawrence, my computer science ULA, absolutely did. And so he really spearheaded kind of helping us figure out how to access that data, dig it out, and get it into R and use it. Because we're trying to figure all of this out while I'm continuously trying to teach the class. So it really ended up being a team effort doing this during the semester. At least every group had at least 1 SNP. They basically came up with a list of SNPs from the literature they were interested in. Not all of them were on the genotype array. You wouldn't expect them to be, but they were able to find something. The electronic health record projects were more successful because we knew what to expect. And we spent a little more because of the genomic data, but it was really a small amount of data, so not much. So these projects, we were back on campus. Everyone was done with COVID. No one cared. So no one touched the COPE surveys this semester. But there was a breast cancer project, one on opioid dependence and abuse and access to healthcare, socioeconomic factors, having an effect on the likelihood of being diagnosed with Alzheimer's, and a project on cardiomyopathy treatments and an association with demographics and socioeconomic status. So the third iteration was this past spring, and we decided we wanted to do a little bit more with the genomic data. And my initial idea was, okay, let's have the students, instead of pick a couple SNPs, let's use the whole genome sequencing data, and let's pick a gene and look at a gene. However, there was a funding call at my university for instructors who wanted to try out new content in that course. And I applied for that funding and got it. So I had $300 basically to play with. Luckily, Akeem was going to be my computer science ULA again. And because he knows the project just as well as I do, we decided we were going to see if we could do a GWAS with undergraduates. And so the idea was that Akeem would work through some of the GWAS sample code in the user support area and kind of figure out what settings we needed, what guidelines we needed to give the students. And we didn't want to spend the students' money for this. So this is what I used that little bit of grant money for. We were completely in person again. And with this kind of project, it was interesting because the design was kind of different and what we got at the end was kind of different. So the students ended up not choosing from all these different data types like they did in the first two iterations. There was a lot of focus on the EHR data and the physical measurements. This group was very, medically minded. And again, we used the whole genome sequencing data instead of the genotype array data. But the way we got out the data and worked with it was similar. And so we still ended up using HALE for that. And it was interesting because these were really ambitious technical projects, much more than we'd taken on before. And it was kind of touch and go, like fingers crossed, we come up with something at the end of the semester. And because we spent so much time on the technical stuff, which was really valuable for the students, what we ended up cutting out was some of the nuance from the previous versions. So the students didn't know their data as well. Their graphs maybe weren't as nice, but they got this whole other technical experience that they didn't get before. I think they're both valuable, but I'm not sure which one I like best yet. We also, with 4 months from not knowing to code at all to GWAS is a lot. And so we were short on time to fully explore the GWAS results. Everyone had a Manhattan plot and could explain it, and that was my goal. Actually, 7 out of the 8 groups completed the genome-wide association study. One of the groups, both individuals in the groups had technical issues enough that we decided that they would just choose some SNPs and work with the SNPs. Again, these projects took more time if the students used more EHR data, and they were actually really worried they were behind. And so we had to talk with them and work with them like, no, it's the type of data you're using. You're really not behind. We've seen this every semester. They spent a lot more money. And the reason it was so high is because I thought the students had, really good technical understanding, right? These kids and their computers now, right? That kind of thing. No, cloud computing was an interesting experience for all of us, right? Teaching them, how it worked when you're running something that's not, this quickie graph you're running in R, when you're actually running, running something in the cloud. What does that look like? How do you check that to make sure it's still running? When it's done, how do you make sure you're still not bleeding money? That took a little time, and I think we still have some places we could grow with that. And that's where a lot of the money came from, is that we would have one person in the group would blow through their money, and then their partner still had $300, and so then we would use that money to finish up the project. So it's not like it really took $600 for everyone. It's just we had some learning experiences there. All right, this past semester, there was a big women's health focus. And just because it's not like I had more women in the class than usual. It was pretty much the same. And so either diseases specific to women or groups of women, say within Crohn's disease. We also had, type 2 diabetes, coronary heart disease, bipolar disorder, a different Crohn's disease project. And the students were, again, really interested and really proud of their projects. And the in-person semesters, we were able to present at a university-wide poster session that they use for students that do independent research and students that are in these cure classes. I'm not the only cure class at my university. We have a really great culture of cures at Towson University that gives students a lot of different research experiences that they can explore. So we use the user support a lot, both with help tickets with the fantastic help team, who has spoiled me for any other kind of help ticket experience because they are so good. And also the user support is where we, if we have a question, how are we going to access, say, the genomic data? That's the first place we look. So with my students at the beginning of the semester, I always have them in class watch the getting started videos, and we kind of talk through them. We also watch the office hours for new users that are recorded and online. I play one of those in class and we talk about it. Written an instructor guide to student projects, which I'll talk about more in a minute. And the two options on here, that one and one of the featured workspaces, have that little blue star, and that's full disclosure / shameless plug. I am author or co-author on those two entries. Featured workspaces that I've used or I'm really interested in exploring more. The skills assessment training notebook for users. That's one that I haven't looked at much and I'm really interested to because I think I want to use parts of that in the fall. There's multiple workspaces, how to work with genomic data, how to work with wearable data. So if you're interested in a specific data type, honestly, my students that were interested in wearables, I always said, great, go look at the user support. And they figured it out on their own. It was wonderful. Data wrangling is a great featured workspace to look at. And we have one that we have written based off of this course. It's based off of version 2, and it's the Genomics Undergrad Lesson Plan Exemplar. And I'll talk about that one a little more in a minute as well. Okay, so in the Instructor's Guide to the Researcher's Workbench, this is an article, and this is based off of a list that I made after the first time I taught this course. And it was kind of, what did I tackle when? So the first thing I tackled was, well, we don't know what we're doing, but that's okay. And the next thing, it's like, well, we got to figure out how we're going to design our experiment. And so this article elaborates on those 10 points, and it talks a little bit about resources you could use, how you can approach it. Obviously, you shouldn't wait till like step 9 to tell your students to keep their code neat and organized. We tell them that very early. But that's, by the time you get to that point, that's when it becomes a problem. And so this was when do I encounter the problems? And this is kind of the order of that. The workspace that we have, as I mentioned, it's basically version 2 of my course. And so after that semester, Akeem and I were like, oh my gosh, we have all this code. It's in random places, like on Blackboard or in, our workspaces. And we have to arrange this and like put it together in a way that we can actually use it. And so that's what we ended up doing. We took the code that we use frequently in the class, put it together the way we've had the students use it, and did a sample project. And it really serves as an example of a full student project. It shows how a primarily R user can access the genomic data. We have little notebooks in the workspace that show you how to pull out the data you need and get it into R. If you're interested in a registered tier project with students, something more like my version 1, You could use this workspace for that too. Just ignore the genomics section at the end. And it really takes you through almost like a tutorial. And we have line by line for the code. And Akeem really put this together based on what we had constructed through 2 semesters of the course. We have exactly line by line an explanation about what you're doing. And you can see how we, arrange the data, how we make the graph. From that graph, what do we do next? What statistics do we do on it? We really use this as a textbook in my course. And so when the students say, we want to, how do we make age groups? We tell them, go look at this. It's all in there. And so that's been really helpful for us because we don't have to scramble and find random bits of code. And so hopefully this is helpful to you as well if you're interested in this type of project. All right, that is all I have for you today. I do want to acknowledge my fabulous students. So the students in this photograph are the students that did the GWAS, and they really were great. I kind of tell students, we're going to do this and it's going to be fabulous. And they have to believe me. And so luckily this group believed me and they bought in. The individual in the little separate picture is Akeem Lawrence, who is here with us today. Again, I could not teach this course without him. He has been really the technical arm, which has enabled me to actually make this course work. I'd like to thank the Towson University Fisher College of Science and Math for really supporting the cures. And a lot of the funding that actually caused Towson to create all these cures came from the Howard Hughes Medical Institute. We had an Inclusive Excellence grant, and we used that money to help get the cures started and some professional development to help us learn more about cures and how to teach them to a variety of students. I got my funding to work on the GWAS analysis from the Faculty Academic Center of Excellence at Towson University. And I have to thank the helpful team at the All of Us research program. Whenever I have a problem, I'm like, hey, can we talk? And I always have a great conversation. And they really are partners and they want the students in the workbench and they really want them to succeed. So it's been really a joy working with them. Thank you.